So today we'll be exploring the three dimensions of the biothreat. After giving you some background, I would like to give you some historical pointers, and then we'll see how genetic engineering, synthetic biology, and the fact that we rely on digital information has modified the way we think of the biothreat. For each of these cases, I will talk about the science and then see how it modified basically the biothreat spectrum, and then we'll see what the science policy has been doing to mitigate the threat. So I think Ray gave you some background. So I was interested in computer language, and I got really interested in the idea that we could treat DNA like a computer language. So that's why I've been doing my graduate research in synthetic biology. And because synthetic biology may be a sensitive subject for biosecurity matters, I interacted with different people in the science policy, and that's how I got there today, and I'm happy to talk to you about my view on biosecurity and bioinformatics. So with that in mind, let's look at a few use of biological weapons. So what I'd like to point out here is, from this graph that I used, is that it appeared all over the years. And there are two cases that I'm especially interested in here. The first one in 1347 is when the Tartars were besieging a city, and at that time there was a Black Plague in Europe that killed 1/3 of the European population. So they took some corpses and sent them into the city, causing the death of several people. And I would like to put that in perspective with the anthrax litter in 2001, where 5 people in the US died after receiving these litters. So in one case, people had no understanding on how disease would spread, and it's only later in the history that we learned the germ theory of disease, and they were caused by bioagents and microorganisms. So the way we think of defense is obviously really linked to the way we understand the life sciences. And I think even from a logical standpoint and the way we talk about biowarfare, we can see that before the germ theory of disease, we really just talk about an effect, which is to cause disease. And nowadays, when we talk about biowarfare, we really focus on the cause, which are the bioagents. For this presentation, I think I would like to present some changes in science. So I think a better definition is really to think of an application of using life science knowledge against other living forms. Try the most current definition, maybe, that comes from the Biological Weapon Convention, and you see that it's also microbial and biological agent or toxins. So toxins are the chemical elements produced by living organisms that are being controlled. And so, to start with an idea of what the biothreat may be, I used this book from Dr. Karras, and he analyzed all the use of biological agents in the 20th century. And he found 180. So by compiling this kind of literature, I decided to present it in this way, where we have bioagents. Researchers have identified dangerous organisms naturally occurring. So it's about a little under 100 of them that are closely monitored. And then they know how people are likely to acquire them, so they can find them in nature. But they can also either steal them or from labs that work with them, or maybe they can be employees. And then there are the dissemination, so can be disseminated through contaminating water, food, through the air, through injections or vector like insects. So that gives you an idea of the problem, and we have kind of understanding of what we have to watch for. And in order to mitigate the threat, one of the policy answers was to create biocontainment measures. So we know there are a few dangerous organisms that are usually kept in these labs that are biosafety level 4, which is the highest level. Anyone working in this lab has a background check and must obey some very drastic safety precautions. So that way, that keeps the bioagents out of reach for people with bad intents. But this has been challenged with the emergence of genetic engineering techniques. In the 70s, we saw the recombinant DNA techniques emerging, and that was a game changer. We were able to take DNA from one organism and insert it into another organism and have it expressed. So early on, that raised a lot of questions among the scientists, and there have been a lot of discussions and a moratorium that ultimately ended in 1975 in Asilomar with an international conference that put in place a lot of guidelines on how to use these kind of technologies. And the risk that people feared was from accidental release, it could be from testing in the open environment, or from deliberate misuse, which is what I'm interested for this presentation. And they are right to worry about deliberate misuse because at that time the Soviet had a biological program and they had secret offensive programs against humans. And from the 70s, they decided to see how genetic engineering techniques could be used to enhance the bioagents. That's research that Dr. Lettenberg and we have been doing, and I would just like to point some of the things that they actually achieve. So Yersinia pestis, which causes the plague, has been rendered able to evade the immune system and also more virulent. Then Bacillus anthracis, which is responsible for anthrax, has been made resistant to antibiotics, which is our current way to protect from it, and so forth. So this program ended when Boris Yeltsin revealed it in 1992. But lately there have been more concern because Vladimir Putin speculated that there could be weapons based on genetic principle, for example. And even though we don't know what that will mean, it's worrisome because over the past 15 years, life science has dramatically changed, and we now see the emergence of the field of synthetic biology. It's about man organism. It's taking principles from engineering and biology to try to study life as we don't know it. So concretely, they will make new biological parts or new organisms, but they may also redesign natural organisms to have either improved functions or a new function that didn't exist in nature before. So the way synthetic biology works, one way to put it is basically we are writing the software of life, and that would be DNA. So here we have DNA at the bottom, and we became really good at writing DNA through the genes and DNA synthesis technologies. Now, over the past 15 years, people have been able to engineer biochemical reactions, and those are working at the pathway level. And so we are getting able to engineer more and more complex synthetic systems. An analogy is often made with computer science, where the early research focused on developing logic gates, and then we have more and more integrated systems. Now we have computers and networks. And I like the analogy because even someone like Bill Gates says that if you want to change the world, you should look at biological molecules. So, like Bill Gates and other people are speculating that synthetic biology is going to lead the third industrial revolution. This is the first synthetic biology plant, large scale, that has been built and that's operating. It was opened in 2013, and here you can see the big farm motors that can grow synthetic organisms and use the products that are made. So our economy is showing signs that we rely more and more on life sciences. The bioeconomy, which includes genetically modified organisms such as crops, that's 2.5% of the GDP in 2012. And synthetic biology alone is expected to be 40 billion of revenue in the next 5 years. And the research keeps going. The UK has just established 3 new synthetic biology centers and is also investing heavily in synthetic biology startups. So all that to do microbial factories, basically. We can engineer this biological system to produce useful components, especially drugs. So one of the success stories, and actually today they actually shipped the first malaria drugs to Africa that were produced by yeast. And this so traditionally, malaria drug is extracting from exotic roots, and it's a costly and timely and sounds not so much process. So they decided to implement the pathway and the way to produce the drug into a yeast, which is much easier to produce and deliver to people who need it. Now, by drugs, I would just like to point out that we can do any type of drug. So scientists have been able to do LSD or THC precursors. And I think some people in the audience may be interested in this kind of misuse of synthetic biology. So similar to genetic engineering, there are many risks that comes with these technologies. And again, I will focus on the deliberate misuse. So let's see one thing. Like this is an example of something that can be worrisome that's done through synthetic biology. So the 1918 Spanish flu killed over 35 million people in Europe early in the 20th century. And so scientists were interested in understanding better this flu strain, so they decided to recover it from frozen body parts that were infected, and they reversed engineering the virus, reconstructed it in the lab, and carry on some pathogenicity experiments. So this led to also the publication of all their results for their research. And this was done at the United States Center for Disease Control and Prevention. But we have to put that in perspective with the fact that synthetic biology engineering is getting accessible to pretty much anyone. So when we publish the information, anyone can reuse it. And we have seen the emergence of a counterculture, so they're called the biobank. There's also a lot of community labs that are going across the world, and one of the things is that they do have a lot of websites where they can publish the protocols and how to set up your own laboratory and develop your own equipment. So with that, this is our synthetic biology and genetic engineering have been modifying the Biothreat Spectrum. We know, see that many different bioagents might be used. It's not only the naturally occurring one, but it could be advanced agents, which are the ones that have been improved for more virulence or be resistant to the drugs that we currently have. There may be also the advanced agents, which means that they are novel agents, and basically I can't really describe them. So we have an affinity of things to look for. And as far as acquisition, manipulating organism is becoming accessible to a large percentage of the population, and they may use gene synthesis techniques to basically obtain the DNA sequence for bioagents that traditionally were regulated and contained into our biosafety level 4 laboratories. So the science policy decided to look into dual-use research of concerns. And that would be trying to figure out if some of the dangerous experiments should be concerned with respect to how much knowledge we can gain from them. So one of the example was early this summer, so that show you how sensitive this is. A researcher took the swine flu, H1N1, and that particular virus caused the global pandemic in 2009, where 300,000 people died. So he decided to mutate this virus until he could render the current vaccine we have inefficient. So potentially creating a new form of the virus that could cause a pandemic. So this kind of experiments create a lot of debate in the science policy community, and in the United States there is a National Science Advisory Board for Biosecurity. In the past, they have sometimes asked the scientists not to publish their data, and then they have organized a lot of debates in the community in order to decide whether or not they should be published. And most of the time, or all of the time for now, they end up being published because there is this strong idea that the data should be shared with the community, so the experiments can be reproduced and so forth. So the current way of thinking is to ask scientists that if they are going to use biological agents that are classified, as well as conducting certain experiments that are considered of potential dual use, they need to get the approval of their institution basically and go through a review process. So basically, we are developing a strong sense of bioethics by putting in place codes of conduct, trying to engage the life scientists to be more responsible about their research. And the best way to put it, I think, is to say that we need a Hippocratic oath for the life scientists nowadays. So, moving on to how people can acquire these agents, there are means to prevent that. So one concern was coming from the gene synthesis industry, and that would be that people might be able to order genomic elements from bioagents that are not supposed to have access to. So the industry, the different companies decided to put in place code of conduct and try to prevent that from happening. And the federal government also published a guidance on how to analyze an order before making it and shipping it. So basically, they ask gene synthesis companies to do two things. One is to screen the customer, and the second one is to look at which DNA sequence they are ordering and make sure it doesn't encode for any of the select agents or toxin. So this is a text document. I had a team of students during my graduate studies, and we decided to interpret this text as an algorithm, implement software that we call GenoGuard, and use this to characterize the government recommendations. So we published our interpretation, and we got the government to give more money to keep developing this project, and also we were able to report back our results to different people involved with this guidance because the FBI was doing a lot of outreach to life scientists who are interested in the fact that students also help mitigating the threats coming from the science community. So I assume you can see that's why I got really interested in the fact that as a scientist I was able to help developing solutions, and that really got me interested, but also I was a bit worried because then I could see that if someone really wants to synthesize organisms, there are alternative ways than commercial gene synthesis. For instance, they could buy their own equipment on eBay, and that got me a lot frustrated, and that got me thinking. And so with that, I was thinking that things are changing in a big way and we need to really understand this. So for instance, until now we were doing biology as a descriptive science where we were trying to observe and understand how things work. And we could observe an anomaly, which was recombinant DNA. We were able to move DNA from one organism to another. And building up on that, we are able to do revolutionary science with synthetic biology. Being in this community, I know that now we are putting a lot more standards and normalizing the process, and I think we are really leading to a new way of making science, or life science at least, and that is we can manipulate them from the inside and we can engineer them to make what we want to do. So with that in mind, if we want to mitigate the threats, not only we need to look at what people are making in the lab, but we should also look at how and what is the process and technology they use to make it. So that's why I got to do my project and looking at the digitization of the life science. And the best way to put it, I'm really happy that people have been surveying synthetic biology scientists and asking them which were the technology that they were using and depending on. And here we can see that the one that were crucial were the public and private registry of biological parts, the standard method for assembling physical DNA constructs, all the genomic databases, all the software tools that we have for search, alignment, analysis, and editing of DNA sequences, as well as the commercial services for DNA synthesis and sequencing. So, just one thing, DNA synthesis is basically writing DNA and DNA sequencing is reading it. So when I look at this, I saw that basically the enabling technology were a lot of data and a lot of robots. So I think it's important to look at what and how each of them work. So I started with data. Where does this data come from? Usually they come from interesting properties that we can find in nature. So this is a jellyfish that you can see at the aquarium, and it's bioluminescent. So in 2008, the Nobel Prize in Chemistry was that they were able to isolate the protein and the coding sequence for this fluorescent protein, and that has been reused in many more projects. So once they sequence and they got the DNA sequence, they put information next to it. So basically, this is the name of the organism, and they say that it's a green fluorescent protein. They have an annotation, a sequence, and they upload that to a biosequence repository. So I say it's open source because everyone contributes. Now, scientists all around the world are able to reuse this information. So if they want to use fluorescent protein for their glow-in-the-dark projects, they may go to these databases, type fluorescent protein, so they will search for annotation, and then they can retrieve a DNA sequence. Then they can cut and paste the DNA sequence and use it. So that's the reference aspect of this biosequence repository. That means that's how people find their information for their science project. Now that you have a sense for the biological data and the biosequence, this is how we use the robots. The field has been developing this bio-design automation principle. So we start with designing biological systems, and there are several computer-aided design tools that have been developed. Basically, they help people to use the data and have a lot of different features to help also doing some analysis and simulation on the computer. Now, people are also able, when they have designed their system, to have the robots making it, and that's called computer-aided manufacturing. And then, once it's done, they can also still use robots to do an automatic data acquisition that will give back to the researcher interpreted data. So, this is how a researcher is going to describe it, but I think it's interesting to see that as a user, this is what you see. You can enter high-level specification, usually through a graphical interface. In the back, there are going to be a lot of data, algorithms, and robots that are going to make your biological system, and then that's what you see at the end. So that's called blackboxing because this, how it works, it doesn't matter to the user. It's just the input and the output that are important. And when I realized that and put it that way, I saw that it was an opportunity for people to carry what I call digital bioattacks. And that means that people could make use of these automated processes to change the data, correct the data, and eventually make more smart reverse engineering processes to disrupt what you get at the end. So one example could be that people, there is many different scales that it could be used. One example could be that people that are against genetically modified research, they may be interested in spamming the reference databases, and then when you rely on your data, you cannot trust it anymore. So that's going to potentially cause a lot of economical damages. Now, if you think down the line in the future, if this bioproduct is a drug that's being produced automatically, and people manipulate it in the way that maybe this drug becomes inefficient, then you're like sending out products that are not what they are supposed to be, and you can cause a real physical harm. So there are a lot of spectrum on what a digital attack might be, but a way we can prevent it would be to look at information security. So there are 3 ways we can think of information security, and to remember it's the CIA, as in confidentiality, integrity, and availability. So confidentiality is about protecting your data privacy, basically. And something related to that would be that hackers are able to discover the name in anonymous genomic studies. Availability means that your system is going to be working and you can use it when you need it. And here we have 2 examples. So the pacemaker have been hacked, and that's why dictionary has it removed, if that can give you an idea of that's a real threat. And that was the same happened for insulin pumps. There are also have backdoors to be hacked. So for these 2 aspects of information security, the FDA, which is the Food and Drug Agency, put in place guidelines that warn these people that are going to develop medical device that use software or that are going to use clinical data, that they really need to take into consideration the issue of confidentiality and availability in their designs. But what I'm really interested in is data integrity, and that's because in biology we have specific challenges. Oh, sorry. It's just before, generally we call it biocrime, which for me is different than a digital bioattack. A digital bioattack has to do with integrity. So this is what I mean by our challenge. If you remember, we have an annotation and we have the corresponding DNA sequence. So in a database you can have the genes that you like to use, and you may also find a gene sequence for toxins. Now, what someone might do, or something that might happen, is that your gene is associated to a different DNA sequence. So when you retrieve it, you're always going to care only about the annotation and just cut and paste what's associated to it. And I think it's an issue that's currently not addressed, and that can be highlighted by the liability and policy on the different biosequence repository. So the first one is the one from the US government. It's called NCBI, that's a big system. So here they do not assume or warrant any responsibility for the accuracy of their data. Well, just let me do a parenthesis. Data integrity is the accuracy and consistency of the data over the time. So here the same, they do, that's the European institution, they do not guarantee the accuracy of the data that they host. This one, I think, is an ironic one. It's an international institution, and they say that the quality and accuracy is a responsibility of the submitting author. So when you are a user, there is no way you can know who submitted the data, so as a user, this is useless. And the biobricks are synthetic biological parts that are used at the undergraduate competition. So they don't talk about accuracy so much, but they talk about safety. They do only accept parts that are not going to cause any harm, basically, or that do not come from any dangerous organism. So I would like to point out that even though these are wants, there are actually no technical solutions to assess it. When I did the DNA screening project, we decided to analyze the Biothreats repository in order to simulate what a gene synthesis order could be. We thought that would be things that people are likely to be interested in getting synthesized. And in all the repository, we actually found at that time the botulinum toxin, which is a select agent. So that's just data, but I just want to point out that we do not really have necessarily the technical means to mitigate the threats. So back to the spectrum, I think that considering all our reliance on digital data, we see that now there are an infinity of opportunity for people to conduct biological data by disseminating maybe erroneous information in databases and so forth. So what the science policy could do could be to really embrace the paradigm shift and not only think about what people are making, but really thinking that since we are going to use so much computers and rely so much on this, we need to realize that what we also need to look at is who is doing what, with which intent, and so forth. So that's why I think it would be interesting to look into a new field which would merge bioinformatics skills with information security to address the specific challenge that we have. So there are a few things that we can learn from generally informatics, and that would be digital rights management. So currently, any sensitive information in biology can be found on the internet. Here you have the recipe to make ricin from castor beans. And also that links back to the dual use research. Right now we publish the sequence of new organisms that have been researched, and a way that digital rights management could be used is to take this idea of a license and say that maybe only licensed scientists would have access to the most sensitive information. And I think that would mimic in the digital world the fact that only scientists that have a background checks are authorized to work with dangerous bioagents. So this might be a way to explore it. Now, something that I care about is that all this biosequence repository that we are relying on more and more, especially as the economy builds upon this kind of data, I think that we should really consider them as a critical infrastructure. And that means that we need technical means to protect them. Some things to explore are being able to identify potentially sensitive sequences independently of their annotations. And this repository have a lot of variety of data, and they are really big, so we need to develop new algorithms to do so. And we also need to think about how to ensure people of their biodata integrity. And here, I do think we have a lot to learn from the banking industry and the internet, where they were able to establish best security practice, give authentication system, and so forth. And I think the first thing we can do and start with is putting in place a trusted third party. So that would be someone in the middle that's able to tell the user if the biosequence they want to use is actually the biosequence they want it to be, and give them this kind of assurance as they are going to use them in sensitive context. And Next, so that would be how to protect our information. Next would be how to defend and prepare for any potential biological attack. And here I think we can learn from microbial forensics. So it's best known for attribution, and we can refer back to the anthrax litter, where the FBI was able to identify which strain had been used and therefore from which location. But forensic can also be used for threat assessment, non-proliferation, as well as verification. So I think it would be worth to think what microbial forensic means in the digital world and kind of have an idea for the signature of the intent and the design of a biological weapon using these databases and using these softwares that we have. So this graph, that's an idea of if someone wants to develop biological weapons, they would start with an intent, then they would design it, then they would make it and test it. And here I wanted to highlight that a lot of the biodefense has been focusing on monitoring for it to be used with like aerosol sensor. Here we have biocontainment and an import-export to try to prevent people from having access to these sensitive organisms. And we also have the DNA synthesis screening more recently. And I think developing methods as digital microbial forensic will be able to try to identify here if someone is looking for sequences and processes and protocols that may indicate that they want to build something dangerous. And in addition to that, that would also help us determine which kind of data really needs to be protected and really identify what are our sensitive and critical information out there. So, let me conclude that I think overall that's not only a problem in biology. I think that in different fields we can see that more and more what I call dematerialization. And that means that people can go around the traditional containment and the physical barriers for people to access weapons. An idea that has been discussed is a 3D printed gun. And actually, yesterday I had a discussion and I thought that was really interesting. Someone said that instead of gun, in biology, it's really about like if we were able to print the bullet. But overall, I do think that maybe in the future we need to look more into a digital footprint for trying to make a dangerous system rather than just controlling the system itself. And this presentation was about Biothreats, so I gave you some historical markers to try to define the biothreat and also say that it really depends on what we understand and how we use life sciences. Then with genetic engineering and synthetic biology, I think that it really challenges in the way that now we are having a potential infinity of bioagents that may be misused, as well as an affinity of way that people can acquire them. And now with our use on digital data, we may have an affinity of way it might be disseminated and an affinity of place to protect. So a way that we could mitigate this would be to set up a field of information security, biosecurity, and really protect the critical resources that we are now relying on. But also try to look from a more defense perspective on what a bioattack might be and as a field of digital microbial forensic in some way. So I would just like to let you read this quote from Lianan, and that was almost 10 years ago, but I think that was really accurate.
I think you've stunned the room into silence.
Okay, and So I'd like to first acknowledge the person that have made it possible for me to do this research this past year, and that would be Dr. Potter, also Dr. Eugen Wolfstahl, and the participants. Also would like to mention that Brian and Ray have given me a lot of feedbacks on this project, as well as Dr. Casagrande, who worked on the DNA synthesis screening guideline, and Dr. Murch, who invented the field of microbial forensic. And so with that, I would also like to thank you all for listening, and I hope you have questions.
I'll start off with the
Okay,
Start with right here. Is there a control here thing?
Hi, so I noticed that you focused a lot on the aspect of biosecurity labs, and I just wanted to bring up the GAO report in 2009 and again in 2013 that found that there were gross problems with biosecurity level 3 and 4 labs where things like anthrax was being carried out in little baggies, and there's just, people aren't taking the regulations very seriously and there's no repercussions for them. Additionally, there's no international standard for biosecurity labs. And since this is something that involves very closely like information systems and relates closely to cyber problems, the biggest issue facing cyber is that it's not just located domestically, but rather internationally. And so you have to consider multiple countries and multiple solutions because each country operates under their own legal systems. How do you see, considering all of this, how do you see the future of regulation, especially well, government and non-state actors, and possible access in labs abroad as well as here?
And as far as the international dimension, I think that's why I was more interested in talking about a trusted third party in the middle, which would be not dependent on the state, but basically depends on the user, and they can use this entity as a reference. As far as lab security, I have not necessarily looked into this. I just used this as a basis to understand how it has been thought of and illustrate that the challenges are that no, the threat also is outside of the lab anyway.
Yeah.
So I guess, do you know if there are any indicators of individuals or groups who have actually had the intent to try to put this into practice? I mean, the theoretical threat is very, very scary. And so I'm just kind of curious, has anybody actually tried to do this, or is this sort of a hypothetical future scenario at this point?
It's, to my knowledge, it's hypothetical. But yeah, I mean, it's interesting in testing for real. I have some ideas on actually testing if it's easy to change the data.
Could I guess? Well, so far this is what you're dealing with, mostly threats having to do with misuse. What about the intellectual property aspects of this? I mean, there could be people out there that using bioinformatics, to steal the information using bioinformatics methods, isn't it? Have you looked at that at all?
For me, the way I've been thinking of the intellectual property issue was with the fact that there are more and more private research centers that do emerging disease work, and therefore they have their own intellectual property and their own knowledge that they produced. And I think that challenges the way we think of biodefense and preparedness, because there is an asymmetry in the knowledge, and thinking that there may be some hackers with bad intents in the relationship, they may have access to information that the defense don't have access to any longer. So the only way I've been thinking of it was more to think that puts we have this problem of asymmetry of knowledge, and that kind of challenge the way that we think that because we have the data we can better prepare, but the data is not open anymore.
Yeah, but you gave some interesting figures at the beginning on what an industry is going to be generated by these some of their, if there's a lot of money in the industry, there's going to be people that's going to be
stealing information.
Yeah.
And that's where digital right management can be explored as a technical way for people to encrypt their data and make sure that nobody accesses them.
Mm-hmm.
Yeah, I'm interested in the biocrime dimension here. In my work, I've never really delved into the bio sphere, if you will. But in my work on illicit nuclear trafficking, I've come across many assertions about the involvement of organized crime, most of which have not really been well documented. There's always been a concern that organized crime will step in and will serve as the kind of bridge between the source and the potential end user. But fortunately, and particularly in the former Soviet Union, fortunately, the financial incentives have not really been terribly obvious. There are other forms of organized crime that have been more productive than dealing in the, or more lucrative than dealing with nuclear material. Yeah, I'm struck. I mean, I know Seth Karras, but I haven't read his book, which has the bioterrorism, biocrime title. Are there really well-documented instances involving kind of biocrime, and how do you foresee biocriminals emerging to exploit some of the things that you've mentioned here?
So as far as biocrime, a lot of it was also considering that people have tried to poison their husband or things like that, and that's included in the book, so that's part of the statistic, using biological agents basically against someone.
But not with a financial dimension, particularly. That's your response is fine, but I was thinking more in terms of biocrimes of a financial nature.
So something I'm exploring too is because in computer science alone, we do see criminalization of attacks. A lot of them are actually just blocking sites and asking money for them to kind of release their data or so forth. So this is, from like being good at doing computer hacking and stuff, I think this is getting a money issue. And as far as who would be interested in, I really think that a lot of activists may underestimate the impact of modifying digital data and might think it's a way to protest without understanding the consequences. And I think,
I have two questions. The first is kind of it's about sort of the current situation. I mean, are these databases kind of just open source? Like, is there anything that's currently in place without the measures that you've already talked about to have somebody who's checking the sequences that are getting submitted? Or is it basically an open source Wikipedia type system where anybody can just send in the sequence and there's nobody to confirm whether that actually is an accurate sequence or not? And then the second is about the incentives that companies might have to start adopting some of the measures that you talked about, like an antivirus to scan for specific sequences. Because it seems like the way that their policies are currently written, they're basically saying the responsibility is not ours. It's the person submitting the sequence and then the end user to make sure that this is actually what they're supposed to be doing. So I guess my question is kind of what type of buy-in might there be for them to actually start adopting some of these measures?
As far as open source, the only ideas in trying to control what gets in is based on quality issues. But the fact that we have synthetic DNA and doing all these synthetic organisms, we cannot assume what should be there. It's open by nature, free kind of information. So people need to register on the website to submit some information. And I'm currently trying to test exactly how it works. But I think they may do homology search, so trying to find if there is the same sequence is already in the database, and that in that way they cannot duplicate the entries. And also they developed a controlled vocabulary that people are supposed to use, so that helps keeping the things of a better quality. But for the incentives, I think, so in biology more and more people are in need to use cloud computing and this kind of private services. So there is actually a company that's called DNA Nexus that raised me once. And they are being bought by Google basically, but they want to provide cloud computing specifically for genomic information. And they have a very extensive data security practices, especially I think one of the main incentives is that they want to ensure their clients they are keeping their data private for intellectual property reasons. But I think subsequently it also helps ensuring their integrity. Even though they're not really focusing on the integrity, but at least they're building a huge wall around the data.
Yeah. First, I want to give you a few compliments because I think this is one of the first few times I heard such a very large scope talk about so many exciting issues which are really in the cutting edge of where we are as society, even as a civilization. So, and you touch with many issues and you are able to connect and to show some connections. Obviously, I mean, you cannot put it much more in 45 minutes talk that much. So I would really encourage you to try to move into something larger, either perhaps to start with a first kind of large, they like the biology from time to time, the format of foreign affairs. So articles, and think about it, putting it as a book. I wish Jonathan Tucker would be alive and he could collaborate with it, because the kind of thing that you may want to have also another author that can complement each other. But I think you touch with issues which are terribly important in a variety of ways, and I think that the lay public would be very much interested in that. And there are not many people who can integrate those issues in such an interesting way. Two specific things. I mean, there is a scenario of the mad scientist, and I think that those kind of technologies allows very much. I mean, Bill was mentioning organized science, organized crime. I think much more, and Jeffrey said about have we seen it? The idea of the mad scientist and the fact that one person or perhaps two tiny, just one individuals on his own in such a way that it's not going to be detected can produce, because the technology to disseminate and to produce and to print is so much available, that create opportunities of crazy crimes and crazy ideas of really demonized form, to the extent that I think we never known before. The other issue that I think I would encourage you to think is, what is the short-term, mid-term, and long-term? Because I think there was a little confusion. What is the far future, mid-future, and early future in terms of feasibility? And the other thing is the analogy with cyber. I think there is a lot of interesting areas of analogy, and I think this is also, which highlight the thing, how much you're touching the cutting edge of technology, of society, and so I really encourage you to continue with that, not to leave it as just one talk, but there are so many interesting areas here. I would truly encourage you to continue with this work.
Yeah, Laura, you talk about this idea of using a trusted agent in kind of the same analogy that we see in the business community. So who would be the trusted agent? Would it be an international organization? Would it be just someone with enough economic heft like Google or something that becomes a de facto standard? Or would it be a regulatory body like the EU? What would be your vision for that?
Ideally, I would have imagined more international institution, non-for-profit, that would
Something like
give this
ICANN
one.
or something.
Yes, but I do not see it coming. I think the industry has much more incentive into starting this kind of and answering this kind of technical issues. So I think an independent company, and I don't think it needs to be something big as Google. I think, and I think it should stay separated. The idea of a trusted third party is that it's kind of independent and it's an authority, so it doesn't have like stake in all of the other aspects of the business. So we start a company.
Doctor?
So a couple of things that kind of synthesize a lot of what people have been talking about. First of all, a lot of your solution focuses on a third-party agent. To comment to that, Craig Venter, the father of the first synthetic life form, he also was really a huge figure in the Human Genome Project. He's partnered with DARPA to create a printer that will print on the molecular level they have a prototype right now, and what they hope to do, and they have done, it's feasible, they just have to engineer it to do it, will print out synthetic life forms, fully just life forms. What they want to use it for is antibiotics, but if you can print an antibiotic, you can print something much more malicious. Considering this, if you have a motivated actor with access to one of these devices, which is not, from the near-term perspective, not necessarily right now, but if this is the future, which is the goal, doesn't that kind of nullify the use of a third-party agent, especially if it's not a government, or if regulations aren't being tightened, since they haven't been, as the GAO report showed? In a way, if you have a motivated actor thinking Aum Shinriko type, apocalyptic millenarian group perhaps, that has access to one of these machines and bypasses the virus scanner, because that's only for if you accidentally print, right? It's bioerror versus bioterror. Then yeah, it's actually, that's a phrase coined by Craig Venter, but it kind of nullifies the use of a third party. If you're very motivated to disseminate something like this. So what do you think about that?
So that's why, if I can go back, that's why I first talk about critical infrastructure and this idea of protecting the information, but then there is another dimension, which is really to try to know which are the sensitive information we need to monitor. And I think if they are going to use a printer, they also need to have access to the information. And if we handle this kind of threat early enough, we are able to know where this information is and try to monitor who is accessing it. And I think that's the way I would think of it.
But you can only monitor so much. A lot of it's already in the public domain, and if you have people who are actively manipulating genes and doing DIY biotech stuff, especially since things like smallpox virus is already in the public domain. Something that's well known is that once a nuclear secret becomes unclassified, it can never become reclassified, at least in the United States. It's kind of that same issue. Pandora's out of the box, the knowledge is there. So even if you can see where the surveillance is, even if you can try and protect that information, and even if you can do some surveillance, it doesn't necessarily prevent a malicious actor, especially if they're using things like in the cyber domain, like the left to right exploit. Like you have your computer set up to look for tag, GAT, whatever, but then they flip it, and so it looks benign when it's really something very malicious. People can be very clever. So I've been also studying a lot about this, and these are the problems I've come across. And I wanted to just pick your brain about it.
No, I agree with your assessment, and my way of thinking about this. Then it goes back to, so there is this dimension of the present and the future and where we situate us. And I do think that anyway, what we are looking at is basically things that we can still find in nature. Moving on, and as we are doing more research on knowing how to design a new pandemic virus and so forth, I do think that maybe we should settle on the debate on dual use knowledge and maybe say that we need to accredit people that are actually have access to this information and at least that would mitigate the risk. I don't know if you, that's the solution.
I mean, I think that's part of it, but I also think part of the whole emphasis on education of life scientists has been not just about, what are the risks of their own research, but also kind of what are warning signs to look at in their coworkers. So you do end up with kind of a Stasi situation where people are watching each other to see what their coworkers are doing. But I think, the more education there is about sort of the inherent risks involved, the more you get a situation of not just self-policing, but co-policing.
If I may, I suppose that when I think of this, I don't necessarily think of it as the vast majority of people are going to be using this technology for good, hands down. But you only need a couple of motivated actors, Shiriko types, Amerithrax, one disgruntled guy who might be able to do this stuff in his basement, probably things up with eBay, buy a printer or something. So I think, yes, a big part of it is ethics and instilling the culture of that, but does that go far enough? Can we police it otherwise?
See, part of the issue of the crazy scientists is that it's very unlikely to be discovered by the radar. It's going to be below the radar.
Yeah.
So it's not something that intelligence would find. If it's one individual, it's very unlikely.
And I think that spans across the CBRN field in general of issue.
Well, thank you, Laura, and thanks for the good questions. And I hope that you come back in a year and tell us what you have accomplished. This is really a fantastic field.
Thank you.